Okay, for students in Form 5, I want to start my part 2 in explaining this chapter 20, the test for photosynthesis, and how to explain the questions accordingly. Okay, so we are up to 11, question 11, part C. Question 11, part C. So with reference to this picture, to this diagram, we want to make sure you know which page to refer to first. Okay, I want you to get to page 34 of your textbook. Okay, page 34 of your textbook, which is shown on the right hand side in this diagram. And this diagram, I want you to explain in the first stage, first period, explain the change in the rate of photosynthesis. Okay, the change in the rate of photosynthesis. Whether photosynthesis is speeding up or slowing down, something like that. Pay attention to the period here, okay, from 0 to 0.01. So in this case, why does photosynthesis speed up when more CO2 is available? Okay, photosynthesis speeds up, okay, or rate of photosynthesis, you can say it speeds up or increases because, okay, when more CO2 is provided, okay, more substrate is provided to Calvin cycle to bind with, more substrate is provided to Calvin cycle to bind with the 5C compound. Okay, if you still remember what is 5 carbon compound, then you will know. Okay, 5 carbon compound, we call that a substrate in this case. So in this case, what is the limiting factor? The limiting factor is the carbon dioxide, the CO2 concentration. Okay, because how do we study the limiting factor? When you provide more CO2, this photosynthesis can speed up. So we call carbon dioxide concentration is the limiting factor. Okay, this is the limiting factor. So on the other hand, part C2, when carbon dioxide concentration increase from 0.03 to 0.04, okay, 0.03, and during high light intensity, the rate increase. Okay, but during low light intensity, the photosynthesis rate does not increase. Why? Because we say in low light intensity, and that is in the blue curve, okay, at low light intensity level, light intensity is the limiting factor. Low light intensity level, light intensity is the limiting factor. Okay, because In other words, when you provide more carbon dioxide, even when you provide more and more carbon dioxide, light intensity is a limiting factor. That means if you don't change your light intensity, no matter how much CO2 you provide, you cannot speed up photosynthesis. So light intensity is a limiting factor, but not the CO2. Providing more CO2 does not help to speed up photosynthesis. I always use this phrase to help you to understand. Okay, providing more CO2 does not help with speeding up photosynthesis. Okay, this is a concept that you need to know when you study the concept like limiting factor. Okay, but how about in high light intensity level? You have to explain the difference in two curves. Okay, explain the difference in two curves. At high light intensity, and under high light intensity, under high light intensity level, when you have more light energy is absorbed by more light energy is absorbed by follicle. Okay. As a result, more ATP and NADPH is generated, okay, to regenerate more 5 carbon compound. Okay, so as a result, more CO2 can be absorbed and the rate of photosynthesis increase. Okay, this is a much longer version than your model answer. Okay, but I hope you still can increase what I'm referring to and know what you should write. Okay. And I clear it. Last one, part D. Suggest one factor. Okay, suggest one factor that you know other than light intensity as is limiting the rate of photosynthesis. Okay, at high light intensity level, at high carbon dioxide concentration, so you have sufficient. The background is that you have sufficient CO2 and also sufficient light. So what may be the other factors? Usually we say it's the number of chloroplasts. Okay, can be one of the limiting factor or the temperature. Okay, so that even if you supply enough CO2, supply enough light, number of chloroplasts or the temperature can be one of the limiting factors. Okay, question 12. The student the student carry out the experiment. Okay, so I've explained the solution of this question paper in during your lesson, and I teach you how to do it. Explain. So first part, explain the result of B. The chloroplast suspension means that they have chloroplast, okay, and DCPIP can change in color when it binds with H. So let me write the word equation first. DCPIP is blue in color. Okay, this is blue plus H. When DCPIP binds with H, you can turn it into colorless. Okay, H means hydrogen. You can turn it into colorless. So, and part A, what's the problem? There's no chloroplast. No chloroplast. In part C, the problem is that there is no light. Okay, so you want question wants you to explain the results of and tube B only. Do not explain the results in other parts. So you only need to explain why the DCPIP in tube B only in tube B is decolorized. Okay, turning it into colorless. Turning it into colorless. So you need to explain why do we produce hydrogen. Okay, first one, chlorophyll molecule absorbs light energy, captures some light energy. If you need to refer to your textbook, you can refer to photochemical reaction. That is page 20. Okay, refer to page 20. Some electrons, when you absorb the light energy, some electrons are emitted. Okay, these are the excited electron. And third part, the energy is used to split the water molecules into hydrogen. You just need to say it's hydrogen. We call this hydrolysis. Hydrolysis. Right? Hydrolysis. That means to break water molecule. Okay? At last, when you break the water molecule, you can produce NADPH. Okay? The NADPH can provide. Okay, you can also write the last one here. NADPH provides the hydrogen to NADPH provide hydrogen to reduce DCPIP into colorless form. Okay, so we call this the reduction. When DCPIP picks up hydrogen, when something picks up hydrogen, we call this a reduction. Okay, second part. When you have some chemical that prevent DCPIP from from decolorizing. So that chemicals actually, let me highlight that chemical. Okay, that chemical may inhibit photochemical reaction. As a result, when you inhibit the photochemical reaction, somehow you cannot absorb the light energy. Okay, you cannot do the electron transport. You cannot bring the energy to photolize water, to break down water molecule. You cannot form NADPH. Okay, just like page 24. When you have some chemical that blocks the photochemical reaction, so somehow photochemical reaction stops, you cannot provide ATP and NADPH, so Calvin cycle also stops. So you cannot produce glucose. Without glucose, the plant cannot grow very well. So this is the diagram that I want to show you. You can write according to the model answer. Okay. Okay, you can say that chemical may inhibit photochemical reaction, so water molecule cannot be hydrolyzed. Water molecule cannot be hydrolyzed, so NADPH cannot be formed. When NADPH cannot be formed, trials 3C compound cannot be converted into trials phosphate. Okay, this is the result when you don't have enough photochemical reaction. Okay, you don't have enough photochemical reaction, you cannot produce enough NADPH, so you cannot produce enough triose phosphate, so you cannot produce enough glucose. Okay, when triose phosphate cannot be produced, glucose cannot be produced either. Glucose cannot be produced for wheat, so wheat growth is inhibited. Okay, so this is 3 marks question. You don't need to write as much as I do, but refer to the logic here. Okay, last part in this question, part C. Name the structure in the chloroplast where the photochemical reaction takes place. Okay, I want you to study the structure of chloroplast. In the chloroplast, we have and this structure for the chemical reaction occurs in the thylakoid. You can call it the thylakoid, or you can call it the granum or the grana. Okay. And granular, you can say they have, okay, provides a large surface area to pack more chlorophyll. This is point number one, or you can say the granular. That means stacks of thylakoid. Granular are interconnected. Granular are interconnected to allow efficient transport of photosynthetic products. Okay, you can write either one. So last question of the test. I hope you can refer to the Kelvin cycle. That is page 22. Okay, right. Page 32, page 22. You need to underline this question, this guideline. Glycerate phosphate is a 3C compound which is reduced to form triose phosphate. That means 3C compound. In this case, we give it a name. We call this glycerate phosphate. We call this glycerate phosphate. Okay, this is a 3C compound. What happens when you turn off the light? Okay, once there's the light off, change. Okay, once the light is turned off, what happens? Okay, you can see. You can check from your model answer. First of all, you have to focus on this part and this part first. When light is turned off, photochemical reaction stops. So you cannot produce enough ATP, you cannot produce enough NADPH. When light is turned off, okay, smaller words. When light is turned off, photochemical reaction stops. Okay, so ATP and NADPH cannot be produced. Okay, this is a very key of your model answer. Without NADPH, without ATP, okay, the glycerate phosphate, which is, we often call it the 3C compound. Okay, 3C compound cannot be converted into triose phosphate. Okay. But the other reactions kept on occurring which includes, for example, from 5 carbon compound into 3 carbon compound. We keep on producing 3C compound. Okay, and also triose phosphate is kept on being produced. No. Triose phosphate may be turned into glucose. Okay, but then you stop one very important part. You stop the conversion from 3C to triose phosphate. Right, you need to write it down. 3C, okay, glycerate phosphate. The 5C compound is constantly being converted into glycerate phosphate. So you have more and more. So you have more and more glycerate phosphate. So 3C compound accumulates. But then triose phosphate at the same time, triose phosphate is constantly being converted into glucose. Okay, so you have less and less triose phosphate. So triose phosphate is exhausted. Okay, or you can say depleted. Okay, from the curve you could understand that curve X, curve X has an increase in concentration. Okay, curve X there's more, so you can see 3C compound accumulates. Curve X is 3C compound, 3C compound, which is the glycerate phosphate. Okay, for 5 carbon for the curve Y, what is curve Y? Curve Y is triose phosphate, it's depleted. So curve Y concentration drops. So we call this curve Y is the triose phosphate. Okay, that's all for this solution. If you have any question, please feel free to ask me.